Welcome everyone to the first lecture of this seminar series in GEOTOP-A, which presumably means geometric topology applied. So I'll be talking about DNA topology, and thank you for joining us. I'd like to begin by talking about a question that as mathematicians you have to answer pretty often is, what is mathematics good for, other than, of course, jobs for mathematicians? And it's the science of patterns. The theory is built on relations among patterns and on applications derived from the fit between pattern and observation. So this is one of the ideas that hopefully you'll see many times throughout this talk today. 21st century mathematics and biology. The mathematics builds models to connect sparse data points, building a continuity of understanding. It's like weaving a cloth. And combined with this is computation, to build algorithms to breathe life into the mathematical theory and to run simulations to enable understanding of these massive biological data sets. Topology, what's that? Well, it's rubber sheet geometry. In Euclidean geometry, you have rigid equivalents of things. You have to take one and translate it to another. So here I've got a circle and two different triangles and a blob. To a topologist, these are all the same things in the plane because you can deform any one of these to any one of the others. The length and is not important for topologists. You can stretch, shrink, twist, and bend, but you can't cut and break it unless you are very careful about how you control the cuts. So we'll be talking about site-specific recombination today, which does cutting and breaking of DNA. One thing that's interesting in chemistry is the enumeration of isomers. This is something Cayley was doing in the 19th century. So here I've got the vertices correspond to carbon bonds, and the edges are covalent bonds between them. And so here I've got N-butane, which has 4 carbon atoms and 3 bonds, and isobutane, which again has 4 atoms and 3 bonds. And as one-dimensional complexes, these are not equivalent. They're not homeomorphic. But I'll be more interested in topological isomers. Here I've got a circle that's living in the plane, nice and round. And here I've got the trefoil knot. So it's moving into 3 space, and here I'm looking at a projection of it onto the plane where you see the circle crossing itself. So this circle on the right-hand side is self-entangled, so it's a knot. And the one on the left is unknotted. You can have entanglement among graphs. You can have circles that link with each other like links in a chain. This is the Hopf link. You can also have entanglement in linear things. Here's an arc that has a knot in the middle of it. But of course, as a topologist, you can thread the ends back through the middle. And you can convert this picture to a straight picture, which is unknotted, or you can have entanglement among things. So you've got to have cycles in the graph to be able to trap entanglement. However, more recently, people are looking at knots in proteins, and proteins are linear. So the question is, if you enclose this knotted part with a ball, then you can talk about local entanglement. So it's a very important consideration these days in the study of proteins. Here are some knots. This is the unknot. This is the plus trefoil, the simplest knot. It's twisted in a right-handed helical fashion. This is its mirror image, the minus trefoil. And this is the figure 8 knot, a 4-crossing knot. This is the unlink. This is the Hopf link, or in chemistry, the simply linked catenane. And this is the Whitehead link, or the figure 8 catenane. And all these knots and links will turn up later on in the talk. DNA is a long and sort of very flexible molecule that can become knotted and entangled. So here's a picture of the Crick-Watson duplex structure, where you have these 2 backbones that are sugar-phosphorus backbones that are wound around each other in a right-handed helical fashion. And attached to these backbones are the bases that form the genetic code. So A is for adenine, T is for thymine, G is for cytosine, and G is for guanine. And A binds only with T by means of a double hydrogen bond, and C binds only with G by means of a triple hydrogen bond. So these things are bonded like rungs on a ladder, and they're twisted in this right-handed helical fashion. And to replicate, a replication fork comes along, breaking the hydrogen bonds, and each of the backbone strands then becomes a template for the original parental molecule. But it's clear that because the templates themselves are interwound, that the daughter molecules will themselves become interwound and give you problems if you need to separate them. Also, the DNA is very crowded in the cell. So this is a picture of the genome for E. coli, the bacterium. It's 50,000 base pairs or so, and all this DNA has to be embedded in this very small volume. And here's a schematic of a chromosome for humans. And the DNA starts out with naked DNA here. It's wound around histones, and the histones get wound around in more and more complicated way, and you finally get chromosomes that you can see under a light microscope. It's clear then that the DNA is embedded in the cells, in the chromosomes, in a very complicated way. And in order for the cell to manufacture proteins, the genes, which are short stretches of DNA that code for proteins, have to be accessed. So somehow the cell has to have mechanisms that allow proteins to get at the DNA in here, to take it out, to make the proteins, and then to put it back. But if you're a topologist, you can see that this right-handed helical structure is a obstruction, a topological obstruction to replication. Here I've got a topologist version of a circular DNA molecule where you see the hydrogen bonds forming the surface, so it's a twisted annulus. And for chemical reasons, it has to have two boundary components. It has to be orientable. So you have the red is Crick and the blue is Watson here. And each one, the red and the blue, are templates. So if you replicate this picture by cutting it along the middle with a pair of scissors, you get two daughter molecules, each of which has the original Crick-Watson helical twist. So how is this problem solved? You see, if you've got circular DNA, which happens in humans and in bacteria, if the cell replicates, every bit of DNA has to be faithfully reproduced, and the DNA has to end up in separate cells. They have to be segregated geometrically. They have to come apart. So the DNA has to pass through itself somehow to make this happen. And it happens by means of proteins. One of them is known as topoisomerase. So it makes topological isomers. So here I've got the red and the yellow correspond to duplex DNA, and the blue corresponds to a protein. The protein attaches to both strands of DNA and holds on to one of them and breaks the other one, passing the first one through and then resealing the first one and releasing it. So it's an enzyme-bridged transient break in the DNA that passes the DNA through itself. So that's topoisomerase. Another thing that happens is site-specific recombination, which sort of exchanges strands. Again, I've got a yellow and red duplex DNA strand, a protein in blue. The protein, again, attaches to two bits of DNA, and it breaks both of them. It breaks the yellow and the red, and it reattaches the yellow to the red. And it changes the genetic code, so you have a recombinant genotype on the right-hand side, and you have a change in the genome and a change in topology that's mediated by this local, interesting bit of topology. And to study this, the idea is it's called topological enzymology. So this was a routine developed in the Casarelli lab in Berkeley in the 70s and 80s. The idea is that you have a protein that you've isolated from cells, you grow it, you put it in a test tube. This is in vitro. All these experiments are done in vitro. And you have a bunch of DNA circles, small DNA plasmids that are circular DNA that are supercoiled. This one has two right-handed supercoils in it. And you grow these circular DNAs, you take a family of them, and you feed them to this enzyme in the test tube, and then you harvest them. You run them through a gel, which I'll talk about in a minute. And you see changes in the geometry. Here I've got two supercoils becomes one, so there's a change in geometry. Here I've got an unknotted DNA that might become knotted. That's a change in topology. And a pair of these things which are unlinked can become linked. So by observing the changes in geometry and or topology, the idea is to deduce what happened in the test tube. So the information you'd like is how much do you know about the substrates? How does the enzyme bind to the substrate? Does it have to see certain kinds of geometric patterns? And then if it's going to do recombination, there are these recombination sites that I'll discuss in a minute. And when it breaks it apart, does it rotate it around to reconnect it? Just how does it do this? So that's the enzyme mechanism. So it's possible to use topology, if you know the before and the after, to figure out what happened in sort of this box where you can't see it directly. So cellular control of DNA topology is extremely important. So DNA entanglement is lethal. It causes mutation and cell death. Topoisomerases, as I said, pass DNA through itself via an enzyme-bridged transient break, and this is used most of the time to fix these entanglement problems. And recombination, which cuts and splice DNA, controls gene expression, and it also can be used to cure entanglement problems. So there are a number of things the cell can do to solve the problems that are generated when the cell has to make proteins and replicate. So the goal, again, is to determine enzyme binding and mechanism. So I'll be talking about site-specific recombination, the Tangle model, an analysis of a particular experiment, TN3 resolvase, and if I have time at the end, about packing DNA in viral capsids. This is a crossover number here. That's a topological invariant. You have these flexible circles in space, and you want to project them down onto the plane in the simplest possible projection. These things, all these twists can be taken out topologically, and these are unknot, so the crossover number is 0. This thing here, you can get rid of all but 2 crossings. This is the best of the minimal kind of projection, minimal number of crossings. This is 2 is the crossover number, and this is a trefoil with 3 crossings. So another very important thing is chirality, handedness. So this is a right-handed coordinate system, x cross y equals z. And if you take the mirror image in this plane, you change the sign of the z-coordinate, and this is now on the left-hand side as a left-hand coordinate system. And here I've got the right-hand trefoil knot, and its mirror image over here is K star is the left-hand trefoil knot. And to get from one to the other, you simply change, you think of the crossings as happening in the z direction. And if you change the sign of the z-coordinate, all the crossings switch. So if you take a knot diagram like this and switch the crossings, you obtain a projection of the mirror image. And it turns out that these 2 knots are inequivalent. There's no way to take one of these and deform it into the other. Another theorem from knot theory is that knots can be decomposed like the integers. If you think of sort of taking one knot and breaking it here and taking another and breaking it, connecting them up, that's the connected sum of knots. And every knot can be factored into prime knots, and a prime knot cannot be factored any further. So here's, this knot is the connected sum of the trefoil and the figure 8. And it's written as 3-1 plus 4-1. 3 is the number of crossings, and 1 is its position in the knot tables. So this is a theorem of Schubert. This is a prime factorization theorem. So it's just like the integers in multiplication. And there are lots of knots. In fact, it can be proven that the number of knots, prime knots, grows like 2 to the n, where n is the crossover number. And the same thing is true for links. So if you go up to 9 crossings, you have a whole bunch of knots. And these knots are, in fact, encoding the complexity of 3-space as being explored by flexible circles. Gel electrophoresis is one of the ways, in fact, one of the most important ways of detecting differences in circular DNA. So here I'm doing, so a gel, this is an agarose gel, so it's a sugar. You can think of it as a random obstruction medium, and the DNA is an electrolyte, so it has a negative charge proportional to molecular weight. And if you put a static electric field on this gel and put DNA in it, then the DNA travels toward the positive electrode. And if the DNA is all exactly the same size, the force is constant on it going through the gel. But the way it travels through the gel is very interesting, because these are knots performed, I'll show you a picture of one of these in a minute, bacteriophage lambda integrase. And these knots are all trefoil knots and TORUS knots, and they travel according to their crossover number. This is the trefoil knot 3-1, I'm sorry. This is the knot 5-1, 7-1, 9-1, so 3, 5, 7, and 9. It jumps in steps of 2 in a gel. And These are twist knots. This is the trefoil knot. This is the figure 8 knot. This is the knot 5-2, and this is these are produced by topoisomerase T4. You get a same sort of banded behavior due to supercoiled knots, supercoiled unknots. So supercoiled travel like this in bands if you have the gel done just right and the voltage right. So you run a gel, and this separates things according to crossover number, and then you'd like to look at them under the electron microscope. And to do this, you can take the DNA, coat it with a protein called RecA, which fattens the DNA from about 20 to 100 angstroms. Here you see in this electron micrograph, you see naked DNA. It's very difficult to detect, for example, what this crossing is right here. But with coated DNA, you can see that this one's over and that one's under. So this is a plus crossing of strands. So with RecA coating, you can detect the knot type on the nose. So here's a picture of a DNA trefoil knot produced by topoisomerase. Again, this is an interesting experiment in the Casaroli lab in Berkeley done in 1985. And it was this experiment where they essentially proved that knots travel according to their crossing number. And this is one of those int knots, bacteriophage lambda integrase. So this is a right-hand twisted plus 13 crossing torus knot. So it's the torus knot 2, 13, twisted in a right-handed way. And again, people study this a lot, but there's a linear relationship. Here's the gel, and here's the average crossing number. And so there's a linear relationship between crossing number and position in the gel. So if you know the position in the gel, you can detect the knot type, or at least you can detect the crossover number. And if you have a reference gel where you know the knot type, you can then argue that the things that you can't see, you can argue about their topological type without actually taking the electron micrograph. Because to do this electron micrograph is a lot of work. It takes a lot of product, and it's hard to do. So the gel is much easier to do, and it's a very, very useful technique. Okay, so maybe I'm willing to answer some questions now about the setup for how the experiments are done. So are there any questions? Okay. So I'll be talking about site-specific recombination. So this is a way, here I've suppressed the Crick-Watson twist, and the DNA is drawn as a line. So all that right-handed helical twisting is not there. And here I've got a piece of DNA with a red site. So that's a short stretch of DNA, so that if you read along one of the two backbones, you see a short word in A, C, T, and G of length maybe 15 to 20 base pairs. And here I've got another site that's the same spelling on another piece of DNA. And these two bits that's the recombination sites, and these two bits are brought together in the presence of this protein somehow. The protein attaches to the DNA in the red and the black, and then it performs a break in the red here and a duplex break here, and the red gets glued to the black, and the black gets glued to the red, and this is sort of the recombined DNA. And to study this, you study it on circular substrates that, again, by cloning, you can make these things and you can install these sites on them. You can change their position on the substrate, and you can change their relative orientation. So here I've got two sites that are in so-called inverted repeats, where if you think about one of the sites as inducing an orientation on the circle, the orientation from the red disagrees with the orientation from the black. So this is inverted repeats. But you can do it with direct repeats, where the orientation of the red and the black agree. Here's a picture of an enzyme bound to DNA. So this is a DNA substrate. You can see it twisted up here. And this is the protein itself that's bound to it. And the sites are down here in this black box. So again, the idea is to use some topology, topological reasoning, to figure out what happened in the box. Because you know something interesting has to happen. And here's the sort of thing that can happen. So here's the substrate shown as a supercoiled DNA. And this is with directly repeated sites. The enzyme brings together the two sites in the presence of this brown enzyme. Reconnection happens, and you see a single circle becomes a pair of circles. that can be linked. Or if you run it backwards, this pair of circles recombines to become a single circle. So if you start with one circle and do a recombination one time of direct repeats, you get a pair. You do it another time, you get a single circle. So it's a mod 2 event. Starting with a single circle, you go to a pair of circles, then a single circle, then a pair of circles if you repeat this process. So these are two-string tangles, and the idea is to use the mathematics of tangles from Conway, where you write down the winding numbers. And if you write down the vector of winding numbers, you can do a continued fraction calculation to give you a rational number, or infinity. And these rational numbers classify these tangles. Again, the tangles can be classified thinking of keeping the boundary points fixed. Because with these rational tangles, if you move the boundary points, then they're all trivial. Then tangles that cannot be undone by unwinding it, moving the boundary points, are called prime tangles. So I'm now switched to, can you still see the picture? Hopefully, yes. Okay.
Yeah,
so,
yes, we are watching the picture.
Okay, great. So it's possible to take tangles and add them together. So here I've got one with 3 crossings horizontally, one with 3 crossings vertically, and then this is A plus B. Or if you like, you could take this big one and cut it down the middle here to give you a tangle that can be subdivided here. So it's a way to decompose tangle into smaller tangles. You can also take tangles and get rid of the 3-ball that defines it by taking the top 2 boundary points and connecting them, and the bottom 2 and connecting them. This is called the numerator construction. And the numerator is related to that rational number that I mentioned briefly. So if you take a 2-string tangle and do the numerator construction, you either get a knot or you get a link of 2 components. Those are the only 2 possibilities. And these are related rational tangles. When you close them up like this, give you a class of knots known as 4-plats or 2-bridge knots. So it's a class of knots that's been studied for a very long time. They're completely classified. So the mathematics is all very well known. So it's possible to take one of these knots and write down a vector that classifies the structure of the knot. This is the knot 6-2. And here's the Whitehead link. And it's got this vector. So this is a way you can use this classification scheme to do the mathematics of the tangle model. Rational tangles and 4-plats are formed by plectonymic interwinding of pairs of strings, which is something that DNA likes to do. So this is probably why this model works. And rational tangles are building blocks for 4-plats, because if you take 2 rational tangles and do the numerator construction, you get a 4-plat, either a 2-bridge knot or a 2-bridge link. So the idea is to use the biology to write equations where the unknowns are tangles. So here I've got a big green box. So think of that as the protein. And it's subdivided using tangle addition into 2 sub-boxes. The one on the left is the part of the configuration that does not change under recombination. It's got 3 plectonymic interwinds. So it's the tangle -3,0 in this picture. And there's the one on the right where I've got the things crossing. So that's the tangle 1. So I've got the numerator construction applied to the tangle -3,0 plus the tangle 1. And the idea now is to forget all the green balls and just think of it as something in 3 space. When you do that, you get the Hopf-link 2. So this is an equation involving the numerator construction on a pair of tangles and a 4-plat on the right-hand side. So the recombination tangles are something that's happening just before recombination and just after. So here I've got this green ball that is the recombinase. It attaches to, the protein attaches to the DNA in kind of 2 ways. One way is it attaches to a geometric footprint, in this case these 3 vertical interwinds, that has to remain fixed. It has to see this in order to be able to do site-specific recombination. And here are the sites called a parental tangle just before recombination. So I call this thing the outside bound tangle, OB, and this is the parental tangle, P. And this thing is the sum of 2 tangles, OB plus P. And what happens in recombination is that the one on the left does not change, but the one on the right does. I mean, something really interesting has to happen. You have to reconnect these guys. So P changes to R. This is the recombinant tangle. So the idea will be to do the mathematics where you can compute OB, P, and R using the mathematics of tangles. So you get a substrate equation. Here I've drawn the answers to this thing. You want to think of these as unknowns. So I've got the protein here, and then I've got whatever else is happening outside. I'll call this the outside free tangle. It's not bound to the protein. Inside the protein, you've got the bound part that doesn't change, and you've got the parental sites. So this is a mathematical construction to write this bound configuration as the sum of two tangles. And so I've got the numerator construction applied to the sum of all three of these tangles. If you forget the protein now, you get the unknot here that with direct repeats. So that's the substrate. Again, I've got OF, OB, and P as the unknowns, and the substrate is something that's known. And this you control in your experiment. You can perform experiments on knotted substrates or on unknotted substrates. In this experiment, I'll be talking about knotted substrates only. After recombination, you have the product. Again, here's the protein, here's the bound part to the protein, and here I had P changed to R. And the outside stuff didn't change either. So here you forget the ball, and now you look at what's happening in 3 space, and this thing turns out to be the Hopf link with linking number -1 in this picture. So again, to run this mathematics, you have to assume that everything is unknown, that all 3 of these guys aren't known. And you know what's on the right-hand side. You do electrophoresis and or electron microscopy to determine the products. And the idea is then to solve these equations for these unknown tangles. So here's the tangle model schematic. You've got whatever's happening outside, which usually you can detect in an electron micrograph of the synaptic complex. You've then got the bound stuff and the P changing to R. And you want to figure out what all these tangles are. So here's what an experiment looks like. So this is an experiment, again, done in Berkeley in the CHIRALITY lab by TN3 Resolves. So here, you do this experiment and run it and then do a gel to separate the products, and then you can run a light through the gel to see how thick the bands are. And from the position of the bands, you can figure out, and then you look at pictures of you take the things out of the bands in the gel and look at them under the microscope, and you can see what the topology is. This is the unrecombined substrate. Again, this is directly repeated sites, so one round of recombination produces the Hopf link, which isn't shown in this picture. I'll show you in a minute. And then you do another round of recombination, and you get the figure 8 knot. So you have 1 circle becomes 2 circles becomes 1 circle again, and then you do recombination one more time to give you the figure 8 catenane. So this is a pair of circles. So this is 1 round is the Hopf link, 2 rounds is the figure 8 knot, and 3 rounds is the Whitehead link. Here on the right-hand side is these links are all prime links, but this one here is a composite link. And you can argue that this product, which is the connected sum of the Hopf link and the figure 8 knot, is produced by the figure 8 knot becoming loose from the protein and then recombining with another copy of the protein, producing the connected sum of the trefoil and the Hopf link. So I'm not going to be thinking about this one. I'm going to be thinking about these 3 products, the first 3 here. Now here's a picture of the Hopf link itself, and it's possible to do a little biology and figure out the linking number of these things, because it turns out that the AT bond is weaker than the CG bond in DNA. So if you put a lot of AT bonds in a stretch of DNA and then heat the DNA up a little bit, you have the bonds break and you have these bubbles forming. So if your original substrate has 6 of these stretches of AT in it and you know where they are and their sizes, upon recombination you can look at the product here, and so you knew where the original ones were in relation to the sites, and here you can visualize these and you can compute the linking number. It turns out to be -1, which is useful because that's chiral information. Chirality is something that's very useful. So here's the Resolves synaptic complex. So here's the resolvase protein. And the outside structure here is trivial in all these pictures. So there's no interwinding between this strand and this one. So the outside free tangle is 0 from this picture. So that's what this says. So there's no interwinding between these strands. So you conclude here that one of the unknowns, you can solve it by looking at the electron micrograph directly. But it's possible to prove a theorem. So the theorem is that you start with the unknot. Here's the equation for the unknot, where O, B, and P are the unknowns. And here's the equation for one round of recombination. You have the Hopf link, and 2 rounds of recombination gives you this figure 8 knot. And it turns out that all 3 of these products are achiral. They're equivalent to their mirror images, if you don't know any linking number information about the Hopf link. And since the right-hand side, if you take the mirror image of the right-hand side, you get the same thing on the right-hand side. That means that you'll have an even number of solutions to these equations. Because anytime you've got a solution, for O, B, P, and R, the mirror image is also a solution. So that's not good. If you're a mathematician trying to talk to a biologist, they're interested in one solution, not 2 or 4. In this case here, I get 4 solutions to these 3 equations and these unknowns. Where the pair OBR is either -3, 0, this is the bound stuff, and R is 1, or its mirror image, 3, 0, and -1. Or another one is 2, 3, 1, another possibility here, and R is -1. So there are these 4 solutions here. An interesting question is, how do you prove that these are the only solutions, that there aren't any other solutions? And the idea is to look at the twofold branch cyclic cover of these things on the right-hand side, because here you've got the unknot. So the twofold, if you think of this happening in S3, the twofold branch cyclic covering of S3 branched along the unknot is another copy of S3. And so what you can visualize this as is doing Dane surgery on things in S3. There's a lot known about Dane surgery, including the cyclic surgery theorem. It's possible to use that and information about Dane surgery on Seifert fiber spaces, and that's how you do this proof. I'm not going to go through the proof today. It's in the paper, though. So how do you get down to one solution, something that the biologists really want to know? Well, you can use this 3rd round product, this Whitehead link, which as an unoriented link is on its own chiral. So the chirality information is enough to get you down to a single solution. So if you plug this in, you now that do 3 rounds of recombination. Again, I'm assuming here that the enzyme binds to the DNA and then stays bound and does 1 round and stays bound and then does 2 rounds, then 3 rounds, always staying bound, never releasing the DNA. Then you get the answer that the biologists believe, that OBR is -3, 0. You have these 3 plectonymic interwinds and R is equal to 1. And it predicts that the result of 4 rounds of recombination is this 6-crossing knot. And fortunately, it's been observed. So this is the 4th round product. Unfortunately, as a mathematician, by the time that Klaus and I got to the biologist and were able to prove this theorem, they essentially already knew this because they'd had a postdoc. They figured that that they drew a model with the pictures. They didn't know it was the only solution, but they figured it was the solution, and they predicted this 4th round product. And a postdoc spent a year looking for it and then finally found it. And then we came along maybe a year later proving that they already knew the answer without knowing this 4th round product, because if you know the linking number, it's enough to give you everything. So what does the Tangle model do for you? It gives you a precise mathematical language for recombination. It allows hypothesis testing. What if this happens differently? It computes all the alternative mechanisms for this processive recombination when you have a single binding event between a protein and the DNA. But the best news is that it can be used with incomplete experimental evidence where you don't do any electron microscopy at all, where you just know the crossing number for the products. Because, for each crossing number, you have a finite number of possibilities, and you can just try them all out with the mathematics and see which ones work. And if there's a questionable relationship between the product of which round of recombination was it and so on. So once you've got the tangle model set up, you can turn the computer on, look at a gel, and let the computer figure a lot of things out. So the good news is that the tangle model actually made it to a cover of the Journal of Molecular Biology. This is an old journal cover here, but that's the good news. The bad news is that people complained about the mathematics in this paper, and it has like 6 authors, and the number of people that read this thing were probably 6 authors plus 2 referees. So, that's the way it goes. But there it is, the tangle model with resolvase happening. So with that, I think I'll stop and ask for some more questions here. That was a quick trip through the tangle model where it's possible to prove theorems about biology. So the proof happens on a piece of paper and on a computer rather than in the test tube itself. And so the real question is it possible to get biologists to believe mathematical proofs? And the answer is maybe. Sometimes, yes, but not all biologists like that. You can't do it in the test tube. It's not happening. So, any questions about the tangle model?
Yes, are there any news about 3-string tangles?
Well, people have worked on it. Hugo Cabrera has done a lot of good work on 3-string tangles. The problem is that 3-string tangles get you into the non-abelian world. And the mapping class group is non-abelian. So with 2-string tangles, everything's abelian, and you can solve the equations. With 3-string tangles, there have been some experiments where you do have a third strand that comes in and it's attached to the protein, but you assume that you can move it out from the protein, leaving you with a 2-string tangle. So the Jairam lab at the University of Texas with Dorothy Buck and Mario Vasquez, and they've done some work on this, and it's possible to do some work, but you somehow have to. You don't do the general 3-string case. You have to move it. You can't do the mathematics. So the mathematics turns out to be too hard. It may be possible to do it computationally. You make assumptions about the maximum number of crossings you can see, and then you try things computationally. So people are doing this sort of thing. But 3-string tangles are just too hard mathematically to prove theorems. But people are working on it.
Is it possible to use this recombination process in the opposite direction, like to untangle or to unlink?
Yes. That's a good question. In fact, there's a recent paper in PNAS from maybe 3 years ago from Mario Vasquez and her group, where they take the result of replication. And the results of replication is you take a circle that, when it gets replicated, you get a torus link, 2-component torus link, a 2n with 2n number of crossings. And it's possible to do recombination, to use recombination to separate these things. So there's a sequence of separation events that happen where a 2n torus link becomes a 2n minus 1 torus knot, becomes a 2n minus 2 torus link, and it gets down to the trivial link, which is what you want, of 2 unknotted, unlinked circles. So that's because for some reason the cell can't do topoisomerase, so it's got to get these circles apart in another way, and it's possible to do recombination to reduce the entanglement to 0. And this is something that also happens in fluid dynamics. You see the same sort of cascade of reduction of complexity from these vortex knots and links through this torus family.
Is there any subsequence of tangle operations that are more recurrent than others?
People have done some simulations on that. Again, looking at band surgeries on knots and links. So Koya Shimakawa and Mario Vasquez have done simulations again, and they And that's the way they did simulations to figure the probability of certain kinds of reconnection events. So in that sense, yes. So that's, but they're doing it on the simple cubic lattice, and it may be that other people have done other simulations too. So that's the one I'm most familiar with. Is again, it's a simulation question. So you have to sort of take what you know about the biology and then sort of do simulations on these things, reconnecting with themselves in various ways. And so this tells you the probability of certain tangle surgeries happening. So that has been done. Maybe Andrei Stasiak and his group, they've done some of these too, I think, with Dorothy Buck. But again, these are all simulation results.
But I think it would be interesting to relate the function or the action of a protein to a specific sequence of tangle operations. If it makes sense, I don't know.
Yeah, again, there are lots of tangle operations that'll get you from any place to any place else. So, well, infinitely many, so you can also make it a lot worse before you make it better. That usually doesn't happen, though, in biology and fluid dynamics, but not unknown. And again, so once you get to situations where you've got outcomes that you really know what they are, you have to do some sort of probabilistic analysis. And a theoretical treatment of this, I don't really know how to do that, but computationally it makes sense.
Sure.
These are all very good questions, and I wish I knew the answer. Okay. So, Carlos, I'm now at 57 minutes into the hour because of various screw-ups with my computer. Shall I keep going? I mean, that's the thing about these electronic...
Sorry, Witt. Yes, please please continue. Maybe another 10 minutes, then a few questions more.
Okay.
Please.
Okay, I'll say a few words about viral capsids. So this is a picture of bacteriophage T4. So this is a virus that attacks E. coli. And here I have a capsid, which is a protein icosahedron, and packed in here is the alien, the DNA itself, which is the signal for the virus. And this capsid is the thing that travels around looking for bacteria. And this is what it looks like when it finds one. It gangs up on it, and there are lots of these viral capsids attached to a single E. coli. And each one of these things acts as a hypodermic to inject the viral particle. In this case, it's a linear duplex DNA strand to inject it down into the bacteriophage, and then it forces the bacteriophage to make copies of itself and copies of this spaceship that it's traveling in until the cell explodes. It blows up, and then these things go out to find other cells to infect. So this is kind of the standard model of a viral infection. Very scary, especially if you're an E. coli here. So here's how the virus attacks. So it's in here, and it comes out here, and it gets down into the cell. This was supposed to move. Anyway, this is the viral particle. It comes down through this pipe and gets into the cell where it's linear, and then it becomes circular because it knows that in the cell there are endonucleases that digest viral DNA. And so if it becomes circular, then it's immune to these endonucleases because it doesn't have any ends. So this DNA is duplex except on the ends, and the ends are single-stranded, but they have affinity for each other. They want to bind to each other. They have the dual spelling at the ends. And so it becomes circular, and then it's in the cell, and then it can choose to either replicate itself or to inject itself through site-specific recombination into the genome of the host. So the DNA is packed in here very tightly. So it's sort of some of the tightest packing known in nature. So if you break with sonication or otherwise, the capsid here, the DNA comes squirting out. And so the idea is to figure out how is the DNA packed inside the capsid. The spooling model is the one that's probably correct. But another possibility is that it just gets packed in there in any old way, random packing. And so the idea is to do some experiments to figure out what the packing looks like. So here's the single-stranded nature. So this is P4 DNA. It's about 10,000 base pairs. And it's single-stranded on the ends. And these single strands have affinity for each other, so it becomes circular once it gets inside the cell. But this can also happen if you take a capsid and destroy the packing here. You get rid of the protein that packs it in there. Then you release these things in this globular state, and then they become circular. They can form links as well, but if you look at the knots, you get very complicated knots. The idea is that once you allow sort of random cyclization in this globular state, you get a whole large family of knots, whereas if you take sort of the same DNA in a relaxed state where it's not all packed in there, you get just trefoils. So without confinement, so this is the effects of confinement on the DNA knotting itself. With no confinement, you get about 3% knots, mostly trefoils. The viral knots that you get with confinement, 95% of them are knots with very high complexity. The average crossover number is about 27. Again, that's detected from gel electrophoresis. We've tried, and it's impossible up to now to take any electron micrographs of these things. So here's the gel evidence from a paper of Javier Arzuaga that looks at the gel and computes, the thickness of the gel. And here you see the crossover number is pretty large. So the average crossing number is about 25 here. It's possible to do a two-dimensional gel to resolve the very small knots. So you run the gel in a single direction down, and then you change the position of the electrodes and change the voltage to higher voltage and run it to the right. And then it takes bands and splits them. So this is the unknot. And this one here is the trefoil knot. It's this blob. But here's the figure 8 knot. Again, these are knots, a reference ladder that you run on the side of the ladder that you know everything about. So this is the figure 8 knot. And it corresponds to this very faint blob over here on the gel. And the 5 crossing knots are the interesting one. This is the torus knot, and this is the twist knot. And it turns that the torus knot moves a little more slowly than the twist knot. The twist knot moves faster on the gel than the torus knot. This is the position of the 5 crossing twist knot, 5-2. This is the position on the actual gel, and this is the position on the reference gel, which is a twist knot. We know that. The thing over here, we don't know what it is, but it's moving in the same position as the torus knot. So if it walks like a duck and talks like a duck, it is a duck. So, if you figure out its position on the gel, what it should be, then you conclude, this is a torus knot without actually looking at it. Hey, this is science. It's not mathematics. You have to make a few guesses, and this is a damn good one. So, okay. But you have this torus knot structure, and here you see for the first time possible composite knots. This is a prime 6 crossing twist knot, and there's either the square or the granny knot. And the two-dimensional gel separates the 6 crossing band into 2 bits, and we believe the bit on the right corresponds to the composite knots. And here you see the possible 7 crossing knots, the 7 crossing twist knot here. The position is a 7 crossing torus knot, and you see this very, very faint thing here. And the reason this one is very faint is because there are very few 4 crossing knots in here. So, again, we believe that this corresponds to the composite knot of the torus knot connects some with the 5 cross or the 3 crossing torus knot connects some with the 4 crossing figure 8 knot. Anyway, it's possible to do this 2-dimensional gel, and then once you know this kind of thing, you can then try to do simulations to agree with what you see in the gel. So one simulation you can do is the one that works in all of 3 space, the pivot algorithm, where you do these crankshaft moves to randomize and piecewise linear embeddings of the circle. And the problem is when you try to do this inside a closed volume, it's very hard because once you do one of these randomizing moves, you end up outside the confined volume. But as you would expect, confinement means higher crossing numbers, and we were able to conclude, I'm sort of out of time now, that the viral DNA is not randomly embedded, because if you do random embedding, even in a confined volume, you'll have lots of figure-8 knots, whereas we see in this one a deficit of the figure-8 knot and the 5-crossing twist knot, and an excess of torus knots. So the torus knot excess favors toroidal or spool-like packing, and we've tried to do electron microscopy, but no one's ever been able to do it. So I think Javier Aswaga and his group now have some money. They're going to try this again, where they're going to really carefully do the electron micrographs. It's possible to do simulations on shorter DNAs. So here's a new experiment that was done in the RocA lab in Barcelona, where you have a 4.7 kb DNA circle. And again, you have a dearth of figure-8 knots. And here's the sort of the knot spectrum when you do this packing and release it from the packing. And we believe that it's possible to reproduce the sort of the spool-like packing. So here's a spool-like packing from a Nature where you do cryo-electron microscopy to get the spherical virus structure. And if you include in DNA-DNA interactions or an apolar cholesteric interaction, you assume the DNA, when it touches itself, has a preferred twist angle, then that reproduces the right kind of picture. So this is without cholesteric interaction. This is with cholesteric interaction, and we believe this is the right picture. And this also produces knots that are sort of torus knots that can be ejected from the viral capsid by simply kind of unscrewing this picture where the knot slowly goes out. So to be infective, these things have to be able to infect. The capsid has to be able to release the viral particle. The viral particle has to go out through this pipe down in the bottom of the viral capsid into the cell. And if it's packed like this, and these are sort of torus knots, they can, in effect, unscrew themselves to reduce their torus knot complexity step by step to be injected into the cell. So the knots are delocalized in this sense. They aren't pulled tight at one little spot, and they kind of unscrew to infect the cells. So there are a couple of papers here. We reproduce the simulated knot spectrum, essentially reproduces the observed spectrum. So we produce the observed surface order, the observed knot spectrum, and here are some references. And with that, I'd like to thank you for listening. So I'm ready for some more questions, perhaps.
Some more questions?
I'm sorry about the picture going away. I have no idea what happened.
Well, I'm curious to know if other than viral capsid, there are DNA knots. DNA knots have been observed elsewhere in other environments.
They've been observed sort of in cells. Again, they are observed usually when the cellular mechanism for fixing them, the topoisomerases, are inactive. So they're produced by protein production. But, these are very small knots. So if you block the action of topoisomerase in a cell, you'll see these things produced in vivo. So, and there's some other places where you can do this on purpose. You can do site-specific recombination in vivo. And then the sites, you have sort of knots and links produced in vivo, and you do site-specific recombination to give you an unlinked product where you have a very small circle. So these are the production of micro circular DNA, like 200 base pairs. So again, this is a way to use knotted DNA in real cells to produce real DNA that's unknotted and very short. So this is now a commercial endeavor in Lynn Ziedrich's lab to produce small vectors of very small DNA. So again, the idea was to use the mathematics to figure out what had to be done inside the cell, and then you know what these proteins do once you've done these experiments in a test tube, and then you can use those. So there's some efforts there. There are other places in cells where you can find DNA, but for really complicated knots, the place to look for them is these viral capsids where they're so tightly packed that you get these incredibly complicated knots. But, you know, if you treat these incredibly complicated knots with topoisomerase in about 3 or 4 minutes, that kills them all. It makes them all unknotted. So the topoisomerase somehow manages to kill them all very quickly. How it knows what to do? Good question.
Well, there seems to be a clear relationship between the confinement and the knot probability.
Yes.
I wonder if
no one knows how to...
is the most efficient mechanism to enforce knotting somehow.
Yes. Yeah. But, you know, and again, lots of simulations. Everybody knows that this has to happen. But in terms of theoretical proofs, no one knows how to attack this theoretically. It'd be very nice to have a way to prove this. But, you know, at this point, the tools just aren't there.
Thank you.
Any other question?
Well, in your simulations, let me finish that question. In your simulation, were you able to replicate the results from experimentation, like the?
Yes. After we added in the cholesteric sort of angle business in there, that helped us. Like get this picture, the correct picture of the surface order, and the correct sort of knot picture. We've got another paper beyond this where we do more of this, but we're much closer to the actual observed what you see in the test tube itself from the simulations. Again, you see these torus knots sort of turning up. These are the ones that are kind of, and the reason for these torus knots is because the DNA itself has this plectonymic interwind. So forcing this cholesteric packing means that you're going to get torus knots instead of the other ones, mostly.
Okay. Yeah. So you said that the DNA has to have the ends connected together, right? The ends of the DNA has to be close.
Yeah. In this
for the bacteria.
Yeah, so it has the cohesive ends, so it's duplex except at the ends, and when it gets injected in the cell, then it becomes circular very quickly. Again, because the cell has the defense mechanism for alien DNA in a cell is to eat the DNA from the ends. But
how can the bacteria, how the DNA can replicate itself? Like, if it doesn't have any ends, how can it replicate? Because you need to.
Good question. It does what's called rolling circle replication. So it replicates the DNA, kind of rolls, and it produces the infinite cyclic cover. You know, it produces like the real line covering the circle, basically. And then another enzyme comes along. So it rolls like, I mean, they've seen this. It rolls like that, and then another enzyme comes along, cuts it into pieces. And then that forces the cell to make these capsids, and then it packs it in the capsids with a motor. Then the bacterium explodes. Scary. You don't want this to happen personally.
Okay, thank you very much.
Good question.
Witt, can I ask a question?
Sure, yes.
This very interesting coming effort of Arsuaga and co-workers that you mentioned about doing a cryo-EM resolution of the DNA.
I think that's what they're going to do, yeah.
Yeah, so that would be very interesting. Would that be for the P4 or for a virus with a smaller genome?
I don't know. It'd be much easier to do it for the smaller one. And, they know Joaquim Roca very well, so if they can get some of it from him, learn how to grow it, or get the stock from which they can grow these things, they'll But I don't know. I mean, I heard this as a rumor, so again, I don't. I hope he does it. He's been thinking about it for years. But, you have to have some money. You got to have a lab. They finally got some money. I guess they just recently got funded by the NSF. And I think this is what they're going to do. Or I hope this is what they're going to do.
Excellent.
I'll keep you posted if I find out. Yeah.
Good. Thank you.
Any other question? Okay, one question, Witt.
Yes.
So do you think that DNA knots are all knots, or can we think about something like to try to characterize DNA knots?
Well, again, some of these knots are sort of indicative of the process that made them, like the ones that these recombination things do. They have a very specific family of knots that they make. Whereas for viral capsids, they're sort of mostly TORUS knots, again, because they're packed in there by an engine, and the engine kind of makes them in this sort of coiled configuration. So again, it's the method by which they were produced that gives you the knot spectrum. So they're characterized in the sense that, you know something about the way that they were produced. And again, from the knots that are endogenous knots in cells, they're usually very small knots, and they're produced, they're like the trefoil and the figure eight and maybe a 5-crossing knot. So these are the ones that are produced in cells when the topoisomerase is sick. It doesn't function like it should. So I don't know if that answers your question or not, but...
Sure, thank you. Yes, Witt.
Another way to try to characterize them is to figure out how they're unknotted. What the unknotting number is on these things. Because if you were to take some of this viral DNA, a whole test tube worth of it, with 30, 40 crossing knots, and, treat it to topoisomerase where it could only do one round of changing. Once it did it, then it couldn't happen again. Controlling this would be rather difficult. You can't... It's very difficult to do this on one knot at a time. It may be, people do single molecule stuff these days, so it might be possible to fish individual knots out and then have topoisomerase attack it and change it one crossing at a time, and you could figure out what happened in the sequence of crossing changes. That might tell you something about these really complicated knots that you get in viruses. But, they're probably mostly big TORUS knots.
Okay, thank you. Is there maybe a last question, please?
Yes. Do you hear us?
Yes.
Can you hear us?
Yes.
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 As you said, this is science, not mathematics. But then
it's a hybrid. It's a little bit of both. Yeah.
Yeah. Sorry. The packing inside the DNA packed inside the virus. You have a model that confirms the observations.
Yes.
Is there a reason, not a mathematical reason, a reason to think that indeed that is the way the DNA is packed?
Well, it turns out that you have to have a motor to pack the DNA in there. The DNA is very stiff compared to the size of the capsid. So you have to bend it pretty well. So you got to have a very powerful motor that exerts force on this thing to pack it in there. And so people have studied these motors, trying to use them for other things. So it's sort of the most powerful sort of microscopic motor known in nature. So they've measured how strong it is. And so the fact that you got this motor pushing this thing in through this pore, this small hole, and the fact that it's very stiff means that once it goes in, it has to kind of try to stretch out again. So it's going to kind of go to the boundary and gets pushed along the boundary by this motor. And as you pack more and more of it in there, then it kind of forces it into the middle as other strands that are getting packed in kind of go around the outside. Apparently, that's what you see in simulations. So, so that's sort of the reason why we think that that's the kind. It's the packing itself that forces you to get these kinds of torus knots out. And the fact that the DNA likes to align with itself in a certain way because it's like a screw. It has this, this helical structure to it.
Okay.
Okay, I think it's time to end this talk. We thanks very much to the Witt for giving us this first seminar. And we thank all of you to attend this first seminar in this seminar held top this occasion. We hope to see you again next time. Thank you. We will stop the recording. Bye.
Oh, thanks for listening. Bye-bye.
Thank you.